Okay, so hi everyone. Thank you very much for the invitation to speak here at this meeting. This is the first time I'm coming to MiFOBio. I'm very excited to actually follow all the talks and learn a bit more about microscopy and science. I know the organization of this meeting is actually quite a lot of work, so thanks a lot to the organizing team. Thanks for fixing the weather. We're going to have a beautiful weather for the coming days, so that's great. Thank you so much. And then, you know, we've all been sitting for few hours now in this room, right? So I'm sure that, like me, you're a bit tired, you know, like you want to go to dinner, you have issue focusing. So I'm going to give you a little break. And I would like you, all of you, to actually stand. Come on. Don't be concerned. And then I'd like you to give your body a little stretch, you know, like move around so that you're ready for the presentation and you can hold on for another like 40, 45 minutes. You know, this is risky, right? Because if you ask people to stand and nobody moves, right? You're left alone on the stage, you know, like you only want to cry. Okay, and so I've been asked to stay in this little corner, which is not what I'm going to do because I have the light, you know, like this is actually not very pleasant. So I'm going to be out of focus. I'm sorry, and probably move on the stage. Right, so let me see if I've got everything. So today I'm going to tell you about how and why plant cells communicate. And let me start with this. Multicellularity has been a big step in evolution that has completely reshaped the Earth's ecosystem and landscape. But for multicellularity to work, we need cells to communicate with each other. They actually need to talk to each other, to exchange molecules so that they can actually tell the neighbor who they are. I'm epidermis. If you're sitting next to me, please don't be. Which state they're in. I'm under stress. I'm under attack. Please prime your defenses ahead of time. So basically, they need to communicate so that they can coordinate action at the multicellular level. And we humans, we have multiple ways of communicating. We can talk, we can draw, we have facial expression. So do cells. Throughout evolution, there's been multiple modules for cells to communicate, direct cell-to-cell junctions, receptor, ligand, synapse, chemical gradient, extracellular vesicles. And what I'm going to tell you about today is how, gosh, they've changed the year, but I can actually run away still. How plant cells have evolved, you know, like fantastic structure to be able to talk, so to exchange molecules between them. So in our lab, we were in plants and we work in this model system, Arabidopsis thaliana. This is basically a weed, right? And we use it because it multiplies very rapidly, so we can actually do genetics more easily. And like us, this is a multicellular organism, right? That needs to grow, that needs to adapt, that needs to actually coordinate action between cells. But plant cells live behind walls. They are surrounded by an extracellular matrix, the cell wall, that maintain the rigidity of the plant body, but prevent plasma membrane from adjacent cell to actually be in direct contact. So I often say they have this non-touch policy. But because plant cells do not believe in living behind closed wall, they have evolved those tiny intercellular bridges called plasmodesmata that kind of compensate in a way this extracellular matrix and cut right through the wall to connect cells together. And you can actually see them here on an electron micrograph. This is a wall, one cell, the second cell, and those tiny bridges that connect the cells together. And this is now another view, right? This is stereo block phase scanning EM, where you see one cell, a second cell, and then at the interface, you have hundreds to thousands of those tiny bridges, basically a membrane tubes that directly connect plant cells together. Here, the wall is made transparent. And virtually every single cell within the plant body is interconnected by those bridges. And they allow the direct cytosolic continuity between plant cells and the exchange of molecules. So if I was to now unleash a fluorescent molecule here from this central cell, it would immediately flow into neighboring cell through plasmodesmata bridges. Now we're very, very far from having a complete picture of all the molecules that move through plasmodesmata. We know there is transcription factor, microRNA for positional information to set up gradients. We also know that we have hormones, sugar also from the photosynthesis for the organs to grow. What we know is that those structures can open and close, very dynamic. They can somehow sense the environment, detect biotic or abiotic stresses, or even developmental clue to modulate the transfer of molecule between cells at a given time during development or in response to the environment. And because of their function in cell-to-cell communication, plasmodesmata bridges have been really involved in pretty much every single aspect of plant biology, and these are a few examples. But now plasmodesmata are not only simple bridge. In every single one of them, you have an ER strand that runs right through it. This is here the plasma membrane, the extracellular matrix, the cell wall in between the two cells, and the endoplasmic reticulum that actually go through plasmodesmata. So basically, an ER membrane tube within a plasma membrane tube. The whole thing embedded within the cell wall. And molecule flow within the space between those two membranes. Okay? Now you can intuitively see how, if you're not capable of regulating those membrane junctions, either the space or, the molecular organization within this cytoplasmic sleeve, you cannot control cell-to-cell trafficking. And oddly, this is actually something that has not been taken account into our field until very recently, because the textbook model actually was proposing that the regulation of opening and closing was occurring mainly from the extracellular space and not from within. So this is the question that we have tackled into, and that was within the frame of an ERC project that is now over. Whether or not membrane contact site and their regulation contribute to the regulation of cell-to-cell communication. And this project actually started with microscopy, when we realized that those membrane functions within plasmodesmata are actually very plastic. So some years ago, we wanted to be able to actually observe plasmodesmata internal organization better than this. So this is how they look like when you're looking in transmission electron microscopy, right? They're very like thin shadow within the cell wall. And this is because they're very tiny, about 20 to 30 nanometer in diameter and maybe like 50 to 100 nanometer in length. So basically in transmission electron microscopy, you fix your sample, you embed your sample, you're doing like thin section of about like 50 to 100 nanometer in thickness. And this is the thickness of our structure, right? So when you image them in transition electron microscopy, you basically flatten the whole structure. You have a 2D projection of your structure and you lose any internal resolution. So with a colleague, some years back, we decided to apply electron tomography to plasmodesmata. So in electron tomography, you're going to now tilt your section at multiple angles. And by doing so, you can now have a view, a different view of your structure. And that allows you to reconstitute the volume of your structure with an increased resolution in X as Y. So you go from a projection to now being able to navigate within the volume and have much higher resolution of your structure. So here, this is the cell wall, the plasma membrane, one cell, the second cell, the endoplasmic reticulum that goes right in the middle. And so we've applied this technology to like many different projects. And one thing that actually struck me at the time was that what we were observing was really very far from the textbook model of plasmodesmata, where the ER plasma membrane gap was kind of fixed, rigid. Because in many examples, what we see is that this gap is actually quite flexible. And this is where we realize that, well, actually, that must be elements that are just at the interface between the two membranes that could potentially regulate this spacing. And what if those elements would also allow the regulation of cell-to-cell trafficking between plant cells? So we went after those elements that are in red here, that we call tether. And those elements we can actually see in electron tomography. So this is here in what I call extended form or compact form. So basically, we wanted to identify them. So what we did was to purify plasmodesmata. We use cell culture that have numerous plasmodesmata at cell-to-cell interface, and then through mechanical description, we can then extract the cell wall, and the cell wall still contain plasmodesmata bridges, right? You can see them here. And then we're using cellulase to remove the cell wall and only to recover plasmodesmata-derived membranes. And then we did that, we purified our structure, and then we purified other subcellular compartments, and we've analyzed them all at once. And for every single protein that was identified, we could actually calculate enrichment ratio within our fraction of interest. So we could now discriminate truly associated plasmodesmata protein from likely contaminant. And by doing so, we've reduced this protein to about 100 proteins. And we went through this proteome, and we were basically looking for proteins that could tether two membranes together, right? And running through this list, the first hit was several members of a protein family called MCTP for multiple C2 domain transmembrane region proteins. And those proteins have everything we were dreaming of. They have a C-terminal transmembrane domain, complex transmembrane domain that we've shown is necessary for anchoring to the endoplasmic reticulum. This is actually a reticulum ER shaping domain that constrict the ER within plasmodesmata. And they have lipid binding domains, C2 domains, that interact with the plasma membrane. Now in Arabidopsis, we have 16 members of this family. We didn't want to actually, generate a mutant with deleting 16 members, so we actually focus on a few members, and we reason that if they are core regulatory element, they should actually be highly enriched at the top of our proteome. They should have been identified across species in different plasmodesmata proteome, and they should be ubiquitously expressed, right? Because I told you plasmodesmata are just everywhere, connecting all plant cells together. And so we focus on 3 members that had, all those criteria. Those 3 members, they express broadly. This is something we've checked by translational GSP fusion in the leaves, in the root, in the shoot apical meristem. And when we zoom into the cell, we see in the localization pattern, these are functional GFP-YFP fusion, so they can complement the loss of function mutant. You see these tiny dots at cell-to-cell interface. This is now another view with lattice light sheet microscopy, where you see those little puncta between cells. Those little dots correspond to plasmodesmata. This is something that we've checked by many means, but also using correlative light electron microscopy, where we can actually preserve the fluorescent signal and have a look at the what these little dots actually correspond to. And those proteins, as far as we can tell, associate all the way along the length of the plasmodesmata pore. Now, if we mutate them, the plant is not happy, right? So if we mutate 2 members out of 3, this is how it looks like compared to a wild type. If we delete the 3, the plant just dies, can't make it after 2 weeks, right? And I'm not showing you the data, but what we know is that those 3 proteins actually work as a complex. Okay, so we had those proteins that we knew was acting at the interface between the ER and the plasma membrane within plasmodesmata. But now we wanted to know whether or not they actually can regulate trafficking, and if they can regulate those membrane junctions. So we ask what happened if we not delete those proteins? What happened to the membrane junction? Is the spacing change, and what is the consequences on the trafficking of molecules? So in the loss of function mutant, what we could see is that we have more frequently detachment between the plasma membrane and the endoplasmic reticulum, which is different from the wild type, where the tethering between the 2 membranes is actually much higher. This is 20 nanometer in diameter. This is something we can quantify. So not only we see more frequent detachment, but the space between the 2 membranes is also larger. So what is the consequences of this detachment on cell-to-cell trafficking? So this we've addressed using a DROMPA, which is a photoconvertible fluorescent protein, where we can, with 2 photons, photoactivate only 1 cell and follow movement into the neighboring cells. And so we ask what happened in wild type, where you have rather tight junction, compared to the mutant, where the junction are loose. And what we could immediately see is that the flow, the molecular flow between cells, it's more leaky in the mutant background. This is something that we can quantify. We measure the fluorescence over time into the neighboring cells. And you can see that in the mutant, actually, they get fluorescence much more rapidly than in the wild type. We see this effect in multiple cell layers within the root of Arabidopsis. This is where we work because this is a transparent organ, so it's much easier for microscopy. So basically, if those protein tether are down, you have more flow. If they're up, you have less flow. So, as a simplistic image, you zip and unzip the membrane junctions. So we know that those tethers can actually modulate cell-to-cell trafficking. But I told you at the beginning of my presentation that plasmodesmata can very easily and rapidly close in response to stress. And so we wanted to test whether or not in our mutant background they could still respond. So what we did was to use different type of stress. I'm not getting into the details, but what happened is that when we stress the plants, in the wild type, the drompa do not move some of the activated cells into neighboring cells, but in the mutant, plasmodesmata cannot close anymore, right? And the molecules actually diffuse cell to cell. Now, I told you at the beginning that these tethers had C2 lipid binding domains. And through molecular dynamics, in vitro, genetics, we actually show that these C2 domains can bind lipids, and specifically anionic lipids, at the plasma membrane. And a category of lipids that are called phosphoinositide that gives the electrostatic charge of the membranes. And so I'm not showing you the experiments, but basically we stand with this new regulatory model where in normal condition, the PI4P level, the lipids, is actually quite low. The C2 domain interact with the membrane, but quite weakly. Now, and then cells, molecules traffic normally from cell to cell. Now, if we increase the lipids level through an enzyme that we have identified, we now favor the interaction of the C2 domain with the plasma membrane. We probably stabilize the tethers within plasmodesmata, and now we lower cell-to-cell trafficking from cell to cell. So this is where we stand. Now we also know that those C2 domains, they also have calcium binding properties. And usually this calcium binding allows very rapid interaction with the anionic lipids. And so this is something that we're currently pursuing. I'm really excited about this because it means that as calcium is a universal signaling molecule induced by many types of stress, now plasmodesmata through MCTP can actually locally decode calcium increase and modulate cell-to-cell trafficking by modulating those membrane junctions. And then we wonder, how those structures actually form, how are they inserted within the wall between cells? And from old electron microscopy studies, we knew that they were actually, or a population of them were actually inserting during cell division. So if you take the textbook model of cell division, it goes as follows. First, you have replication, and then you have mitosis, so where the genome are split between the daughter cells. And then you have a step called cytokinesis, where gradually the two daughter cells are going to build a separation wall until a final stage called the final cut or abscission, where all membrane and cytosolic continuity is lost between the daughter cell. So you have to imagine, you and I, we share this room, right? And we want a bit more privacy. We want to be able to organize our own space the way we want. We just built a separation wall, physical separation wall, to split the room into two. Now, plant cells also undergo cytokinesis and cell division and cytokinesis, but here they build the wall from the center of the dividing cell that expand towards the air, toward the edge. And when they do that, they do it incompletely. So they actually leave holes within the separation wall. And those tiny holes, here in black, are plasmodesmata bridges, right? So if I'm taking this analogy I've made earlier on, right? We build this separation wall, but now I'm going to leave some bricks out. And through those bricks, we can now communicate between the two rooms. So I like to say that plants fail cell division, but of course this is not the correct term. They actually undergo a process called incomplete cytokinesis to build up the intercellular bridges. And this is actually a strategy that we found in multiple clade in the tree of life, and especially in animal cells with specific cell type, the germ cells, that undergo incomplete cytokinesis to form one single bridge between the two cells. And here it progresses as furrowing until the process is actually stopped. But plant cells, as soon as they divide, every single cell will produce hundreds to thousands of plasmodesmata bridges. So we basically wanted to understand a bit more about this process, which is how to connect and divide at the same time. So during this cytokinesis, when the plant cells build the separation wall, they build a structure that is called the cell plate. And the cell plate is a precursor of the cell wall, right? Plus the plasma membrane. So it takes the form of a disc-shaped structure that expands as division progresses. First in the form of a tubular vesicular network that expand, mature, and this structure contains little holes called fenestrae. Those little holes, some of them are going to be maintained and form plasmodesmata, and others are going to be sealed off. And so we wanted to actually understand where is the decision coming from, whether to keep or to seal. Now, I told you that plasmodesmata contain endoplasmic reticulum. And so if you actually image the endoplasmic reticulum across several cells, you actually realize that plants have one single physical ER compartment that stretch over several cell layers, right? And this endoplasmic reticulum running within plasmodesmata to connect cells actually is present as soon as plasmodesmata form. So this is plasmodesmata at the end of cell division, right? You see here in electron tomography. And every single one of them do have already an ER strand running through it. So we wonder how early is this association of the ER with the forming cell plate that would become the cell wall. So we've traced back the endoplasmic reticulum dynamics with that of the cell plate formation. And you can see that as early as the cell plates start to assemble, the ER actually start to accumulate within the cell plate plane. Not only accumulates, but actually stretch through it. So this is Airiscan confocal imaging, where you see the cell plate in pink. The structure is building up. This is over time. And you see the ER strand that looks like as running through the cell plate. And this is something that you observe much better and easier if you're doing like electron tomography. This is here STEM, where you see the cell plate vesicule that are going to fuse and assemble to form the cell wall, and the ER actually running through the cell plate. So as soon as you start building up this separation wall, you already have the ER stretching through, okay? And I told you that this cell plate has little holes, right? And those holes are going to become plasmodesmata or being sealed off. So we started to wonder what is the relationship between the fate of those tiny holes, the fenestrae, and the ER. And so for that, we've imaged dividing cells at different stages. We've captured many fenestra events, little holes. And we did that using electron tomography, where we've imaged through entire lengths of the dividing cell plate. And so we could actually now look and assess, capture the ER in relation to the fenestrae, whether it's going through it, whether it's continuous, associated, not associated. And we looked at two stages, end of cell division and very late cell division, and very end of cell division, okay? Every single dot is one fenestrae. And to start with, the fenestrae are very big, and then they get smaller and smaller. And during this process, the ER is either running through, continuous, or fragmented, or even sometimes absent. Now, as the fenestrae gets smaller and smaller, all of a sudden, you actually have only events where the ER is actually continuous through the fenestrae, very much like what we see after cell division in mature plasmodesmata. And you have a population of fenestrae that are even tinier, but those ones never have ER associated with it, right? Never ER running through those fenestrae. And what we've noticed is that only the one that contain continuous ER are actually found in the final stage of cell division. And those ones, presumably actually close up, are sealed off. But the ER alone cannot do that. So we went back to our proteins, MCTP, tethers, and we saw that, well, they might be indeed regulatory element, but they could also be structural elements, right? And so we look at them, not in mature plasmodesmata, but during cell division, during the formation of this cell plate. And we can see that those members I told you about, 3, 4, 6, associate with dividing cells. Ooh, I should run again. Okay, this is a tiny a red arrow. And during this process, the proteins are associated with the endoplasmic reticulum until one stage at the end of cytokinesis, where the protein becomes enriched within plasmodesmata. So then wonder, okay, they are here at the right time, right? But do they help forming plasmodesmata or stabilizing plasmodesmata formation and promoting incomplete cytokinesis? So what we did was to actually map plasmodesmata and count them, and we've used our serial block phase scanning EM for that. We can image through several cells layers within the roots, and then we can very easily see plasmodesmatas, tiny black lines, and then basically count them on dividing wall. And we can also, so all the tiny red dots that you see are plasmodesmata. And so basically we can very easily distinguish just morphologically late cell division to end of cell division. And so we counted plasmodesmata in the loss of function mutant. And what we found is that we have about 50% less plasmodesmata with continuous ER at this stage, which end up with about 50% less stabilized plasmodesmata. So what this is telling us is that the ER set the balance between abscission or stabilization. If you have fenestrae with no ER inside, or the ER is not being stabilized and maintained, you actually close the fenestrae. If you now maintain the ER inside, with the help of MCTP and probably other proteins, you now actually stabilize the structure and you form plasmodesmata bridges. I have one last story. Are you still with me? Good. Okay, so this is unpublished. And I told you that there were a lot of different molecules that traffic through plasmodesmata. And what we wonder was whether or not lipids could actually move through plasmodesmata. Okay. Now we know that lipids are fundamental element of biological membranes, and that within a cell, you have lipids trafficking through vesicular or non-vesicular traffic through lipid transfer proteins to establish lipid gradients. So not all organelles have the same lipid composition, and this is very important to actually set membrane territories within the cell, with one membrane having a different properties like electrostatic properties to another organelles, and that regulates many cellular processes. So internal lipid gradients. But now we wonder, what if this lipid exchange or this lipid gradient, lipid exchange, not within the cell, but not within cell, between cells, could now establish lipid gradient. Because basically what we have here with plasmodesmata are those hundreds and hundreds of membrane tubes with plasma membrane and ER being continuous between the cells, right? So we wonder whether or not lipids could basically flow within the plane of the membrane from cell to cell. And this is a question we've tackled with a wonderful friend, collaborator, and amazing scientist, Emmanuelle BAYER, who is working in Lyon, and a postdoc, Emmanuelle BAYER. So can lipids diffuse from cell to cell? So what we've used first is fluorescently labeled phospholipid. We've used top fluorPC that when we feed the roots, actually accumulate within the endoplasmic reticulum. And if we frap this fluorescently labeled lipid, we see movement from cell to cell. We've checked that it's incorporated into the membrane, that it's not being, like cleaved off or whatever. I'm not showing you this data. And it actually moves slower than a cytosolic dye, which makes sense, right? Because here you within the plane of a membrane and not in the cytosol, so you expect the movement to be slightly, slower, but faster than ER-anchored protein, which at this time scale do not move cell to cell. But does it really move through plasmodesmata, right? This is, you can imagine any sort of, vesicle secretion or whatever being taken up, So this is actually a very difficult point to tackle. Oops. Okay, what we actually argue was that if this lipid diffuse through plasmodesmata, it should move more through walls that have plenty of plasmodesmata, right? And so we took advantage of the fact that in the epidermis of Arabidopsis cells, root cells, we have way more plasmodesmata in the apicobasal wall than in the lateral wall. This is something that we can quantify. And then we've used fluorescent loss in photobleaching, where we repetitively photobleach a cell and look at the fluorescent loss into neighboring cell. And any neighboring cell that are in direct contact should lose fluorescence. And because now we have more plasmodesmata in the apical wall, we should actually see loss of fluorescence in new neighboring cells that are in this direction. And so this is exactly what we see. But now what I'm showing you here, okay, it moves through plasmodesmata, but is it moving within the plane of the membrane, or is it moving because it's being taken up by a protein carrier that actually move through the cytoplasm? So to distinguish between those two options, we've used MCTP mutants, because those mutants have less plasmodesmata, so they have less membrane tubes available between cells, but they show higher cytosolic movement of molecule because the membrane gap is actually loose, right? So if the lipids move along the plane of the membrane, we should see less movement in our mutant because there are less membrane tubes available. If it moves through the cytosol, we should see more movement because we know that cytoplasmic gap is actually larger and cytosolic molecule move faster. And so what we see is actually less movement, suggesting that yes, indeed, it's actually moving within the plane of the membrane. Okay, good. What about other phospholipid and endogenous lipid, and why is it, what does that matter at the end of the day? So for that, we actually focus our attention on another lipid, phosphatidylserine. For several reasons, we can visualize them within the cell. These are tools that Yvon Jaillet's lab have been developing. So through a sensor, we can actually recognize the phospholipid. We can actually image where PS is within the cell. And we also know the enzyme that synthesizes PS. So in the wild type background, the sensor goes to membranes because those membranes have PS. Now, if you stop PS synthesis, you have no PS in the membranes anymore, and the sensor is now cytosolic. And so we thought we're going to combine those two to design a genetic strategy to indirectly probe PS movement from cell to cell. So if we express the PS sensor within the loss of function mutant that do not produce PS, the sensor is cytosolic. No PS into the membranes, sensor says stay in the cytosol. Now, if we're capable of expressing within this loss of function mutant the PS synthase, but only in some cell type, right? In those very cell type, we should recover the membrane association of the sensor because PS is being produced, and so therefore the sensor is going to associate with the membranes. So we wonder what happened to neighboring cells. If PFs move, we should actually see the sensor in neighboring cells that do not express the PS synthase. So those cells do not produce PS, but the sensor should still move to associate with the membranes. And so this strategy is actually just very easy to achieve in Arabidopsis, especially in the roots, because we have this radial organization and plenty of cell type specific promoters. So we express first in the central cells of the roots, the PS synthase within the PSS1 mutant background. So we only produce PS in those cells and wonder how the sensor looks like into neighboring cells. And when we've looked, to our amazement, every single cell, neighboring cells, actually show the sensor at the membrane. So it means that PS from the site of production here has moved cell to cell to neighboring cells. And this is something that we've reproduced with different cell type specific promoter. Now, why does it matter? So PS is present in all cells across the root. But when you have a look at the expression of the PS synthase, it's actually confined to the epidermis. So PS is not made everywhere within the root. If you look into more details, you actually even have a gradient of expression of the synthase from very strong in the elongation zone to weaker and weaker until you have nothing. We can quantify this. And so it means that we have zone, even within the epidermis, of high PS production and zone of no PS production. If you see how this translates into the pattern of localization of the PS sensor, so where PS is going to be localized within the cell, we see different localization. We see endosome and plasma membrane where PS is produced, and mainly plasma membrane where PS is not produced. And this gradient was actually known before. So this is again work from Yvon's group. This gradient of PS across the epidermis is known to regulate the signaling of Rho GTPase RAP6 in response to a hormone called auxin, doesn't matter, and downstream signaling, and especially gravitropism. So we knew this gradient was important, but we had no idea how it was actually made. So what we think happened now is that when cells produce PS, we have endosome plasma membrane. When cells receive PS from a source, it now favors plasma membrane only. And we can actually check this by only expressing the PS synthase in the epidermis. We have the sensor, the PS, that is endosome and plasma membrane. Now, if we produce PS in the next cell layer, but look at the PS sensor in the epidermis, so PS has moved, we have mainly plasma membrane localization. So we're very excited about this story. So we think that implant plasmodesmata function as a continuous lipid distribution pipeline that allows to actually set up lipid gradient that are important for cellular and signaling function. And I'm going to stop here. The two stories, this lipid movement and the membrane tethering story, has been done between our two labs, my lab in Bordeaux and Yvon Jaillet's lab in Lyon. This is two wonderful postdocs that have been doing the work for the membrane tethering. The cell division was done by those three guys, and really like a big thanks to Patrick Klee, just amazing PhD student. Early career scientists, thank you. You know, like we can't do anything without you, so thank you for working so hard by our side. This is really important to acknowledge, you know, who is really doing the work. That's them. And in Bordeaux, we have the chance to have a wonderful imaging platform that goes through electron, photonic, and also have plant imaging expertise. And my group is using this platform a lot. We are very lucky. These are the people that have been specifically involved into the project I was showing you, but we collaborate with many more. And we have wonderful engineer that really works by our side to help us to actually ask scientific questions. So, you know, if you ever want to study and work in Bordeaux and do imaging, this is the right place to be. And I'm going to finish now by a little movie to show you how much we love our PhD students. So this is a movie that we made for the PhD defense of Patrick Lee, the guy who did most of the work for the cell division paper. So remember, cell division, the ER accumulate in the cell plate and go through. With a French song, of course, you know, like... And so I hope you can recognize the ER strand. The nucleus is actually reforming between the two dividing cells. The vesicles come in from the cell plate, and we gently fuse within the, you know, like the central plane to form the cell plate and the cell wall. The ER strand is in between, and sometimes it actually doesn't hold. It would break through, and if it breaks through, then the fenestrae are going to close by and no plasmodesmata will be formed. And with that, I'm going to finish. I thank you very much.
You mentioned translocation of lipids, also of DROMPA, so small proteins. Mm-hmm. So do large proteins translocate? So does RNA translocate? So that would be cool for expression of you know, genes through these cell
So
walls.
we know, yeah. So we know that microRNA actually moves cell to cell. There's been like indication that messenger RNA might actually also move cell to cell, but these are huge. And those conclusions were obtained through grafting experiments and collecting like your phloem sap, so the vasculature. And this has been actually a lot of debate in the field whether or not we could actually trust this data. But that seems that in some instance, the messenger RNA moves along the proteins. But that seems to be also a size limit, you know, like to